Okay, welcome to the first lab on principal components using R. I want to begin by first clearing out the memory, and I'm going to just do a graphics off because I was running a few things here um, before this lab. So here we're going to load um, two. I think we can get away with loading just one package. The package is this Conan package. This is the package that you don't need this for principal components, but this is the package that will allow us to do these self-organizing maps. And I'll talk about that perhaps after principal components. I might have to take a break and put that in the next segment. Now, what we want to do here is we want to call in some data, and the data that we're going to use is called wines. And this is a famous data set. And it just so happens that it's part of many packages, but we don't have to load anything specific. Oh, actually, here it is in the Conan package. It's also part of the MMST package, which we used to use a lot. For some reason, that package has not been updated in a while. So here we are. We have a wine data set, and it has 177 types of wines, brands of wines, and we also have stuff for that talks about the different characteristics of the wine. And this data set is used a lot in classification because there are different kind of types of wine. I'm not a wine expert, but let's just take a look at the data. Head wines, okay? Now, unfortunately, they don't do a great job describing the different types of wine or types of variables here. But if you look, for example, if you just Google wine dataset, you can certainly find this dataset. It's one of the most popular datasets on the UCI machine learning repository. You can get the information about these different variables there. There's not a ton of standards for what you have to kind of report. Here, I'm not sure if this will tell us anything. Okay, it tells us, it brings us right to UCI, and it doesn't tell us much about the variables. It's not for this specific data set. But let's see. Anyways, you can find that out for yourself very easily. Otherwise, see me if you have problems. These are different characteristics or measurements for the different wines, and the objectives are kind of different. Here, we're going to be thinking about this as an unsupervised problem, and so we can see that if we do dim wines, that we have 177 variables and 177 samples and 13 variables. Now, this is not necessarily a high-dimensional data set by any stretch of the imagination, but still, there's always benefit in kind of breaking this down and looking at things in terms of principal components. And this one I know we're going to look at it with respect to the self-organizing map, so why not start looking at it in terms of principal components as well. So here, what I want to do is I want to first do just a box plot. Now ordinarily you'd do other kinds of diagnostics on this data first, but I just want to look at the data, and we can see here that we have an issue of scales. And this is part of the reason why I picked this data set. So, everything here is kind of quiet and small, relatively, but we have this big proline which has very high levels just by the nature of the measurement. And so that's actually going to be important later. So this is something that we want to keep in mind. And I'm going to introduce you to two commands here for doing principal components on the data. And so let's see here. Okay, The first one is going to be Prince Comp, and I'm going to just pull this up because I want you to see that there's actually two different ways to perform principal components in R. This one is the one that actually does the computation using the covariance matrix of the data. Now, and we've talked about that specifically. Here it is. This is the details. The calculation is done using the eigenvalue decomposition of the correlation or covariance matrix. Now, this is actually how we derived it, but what I didn't mention to you, but it is important to keep in mind, is that that's actually a very unstable way of finding the principal components. And let me give you some idea of why that's true. First of all, correlation and the inference of a covariance matrix in a high dimensional data set is a tricky business. So there's that estimation based on the samples of the covariance matrix. Okay, so if you have a small sample size and you have a ton of different variables, that can be an unstable inference. So that shouldn't be too surprising. The eigenvalue decomposition, there's less issues with that specifically, but it's actually the inference of the covariance matrix that is the problem. Now, for those of you that have had linear algebra or numerical analysis, you know that there's a relationship between eigenvalues and singular values. And in particular, just a note on the SVD, every single matrix has a singular value decomposition. So it's much more flexible in terms of a decomposition than an eigenvalue decomposition, for which you need to have a positive definite symmetric matrix. And there's a relationship between the eigenvalues and the singular values themselves. So the singular value decomposition, it also has a matrix form, and it has a matrix kind of in the middle is the middle factor, which holds the singular values on the diagonal. And just like the eigenvalue decomposition had the same kind of structure. And there is a relationship here between those values. So it's not surprising that you can actually derive the principal components using the SVD. And that's done in practice as a more stable way. And in fact, in MATLAB, they only consider using the SVD. They don't consider using the eigenvalue decomposition in the actual computation. This one, so this exists, okay, and so we can use this. So we certainly have a big enough data set and not so many predictors. So we can use this, but I'll show you we're not going to use it in the end anyways. But just to show you that it works the same way. Okay, and a lot of people I know have actually been outright angry because this is still available in the R package. Because without knowing and without reading the help carefully, you don't really understand that it can be unstable. So we can do a principal component analysis using Prince Comp, okay? And if I do names PCEX1, I can see all of this information here. A lot of it that we have seen before. So the most important things in this particular case are the scores and the loadings, okay? And then there's information about the scale, the number of observations, the centers, the standard deviations. So one has to look carefully and see that you can actually set, so here you can give it X, and you can set information here. Let us see here. Okay, the scaling, it doesn't look like you can scale internally here, but I might be mistaken. Okay, so that's one example, and I'm going to go through two more examples before we actually look at some of these things. Okay, so here we have PR comp. This is the other one, okay, and PR comp, I'm going to do it to the same data set, PR comp. Okay, And so here we have more specific information. We can't actually center the data and scale it, which we need to, at a minimum, center our data. Um, we typically also center and scale. We typically do what's called like a z-score. We subtract the means of the columns and divide by the standard deviation. That's pretty much standard. And the issue, if you don't do that, you can see right away what happens, and I'll tell you about that in a minute. So that's one example, PR comp. And what I'm going to do here is I'm going to say, let's center the data and let's scale it too. Okay, so dividing through by the standard deviation, centering the columns, dividing through by the standard deviation. And then I'm going to do a third one actually. And in the third one, I'm going to use the function scale with PR comp and not do any of the other settings. So scale is a function, and by default, it will center and divide through by the standard deviation. So the default, it will take the mean of the column and center it from the different columns and divide through by the standard deviation of that column. You can actually, by saying true, you agree to do the mean, but you can specify a centering value if you wish. So there's some flexibility here in doing that. You can use this function within PR comp and then just leave these pieces alone. It's totally up to you. Okay. And in fact, what I'm going to do here, and no, I'm sure somebody can correct me on this, but I'm going to say center is false and scale is false. It doesn't hurt just to make sure you're not double scaling and standardizing. So I'm going to run all three of these actually. Okay, so all three have run. You saw that it was very fast. Now let us look at the results. And so actually, I'm going to look just plotting the object, and you'll see kind of what the default plot is. I'm going to open 3 windows to visualize these more concretely. PCEX1, plot PCEX2, and plot PCEX3. Okay, so let us see here the first one. Okay, this is what happens if you do not scale. Now I said that Prince comp was unstable. Okay, it is unstable, but it's not unstable for this particular data set. It has enough samples and few enough variables to estimate the covariance reliably. The problem with this is that everything, if we look at the variances relative to the other ones, you might say, oh, this is great. This means that we can really, this is a scree plot, okay? It doesn't do a great job of the labeling here, but it says that this is the variance for the first component, the second, third, etc., all the way to 10. Now you remember what we were looking for was this elbow. So the elbow certainly exists, but it's an artifact. And whenever we see a huge, huge spike in the first component and everything else is basically nothing, we know that there is a scaling problem. And that is what this is telling us, often misread, but just be wary of that. All elbows are not, created equally. This is something that is in fact an artifact. And so let's see if that goes away. Oh dear. I better save this just in case. Okay. Okay, let's see if this goes away in the second one where we did scale. Okay, here we are. This looks more reasonable, like something that we are used to seeing. Here we have kind of 2 principal components that are big in size, and then we see this tapering off here. And if we did the same thing with the third one, I'd like to think we're going to get the same exact result because we just used a different function, and we have. So we have the same result. Just looks different because one's bigger than the other one. So we get the same result using PR comp with the scale routine or just doing it within the function itself. But it's obviously more advantageous to use PR comp because of the stability, but what we see here, we also confounded it with the scaling problem. We see that when we have a box plot like that where we have things in different scales, um, centering and or standardizing becomes way more important. And even where there's subtle differences, you will see that it becomes important in practice. So that's that's the plotting. Okay, now I want to show you, let us see here. I have some notes here. Let's look at a summary of PCEX3. Since these are the same, we might as well just take the third one. And here we have the importance of the different components. The most important things are this column here and this column here. So there's a couple ways you can think about this. This is that picture I showed you with the scree plot, and you can also think about kind of its complement, which is a cumulative description of the percent of variance explained. Here, if we were to think about the proportion of variance, we can see that the first one explains 36%, 20%, then 11, then 7, and so forth. So that's a pretty big jump between, almost 20% and 11%. That's a pretty big jump. And so that kind of a jump, you might say, we would retain the first two principal components, which is a huge advantage. So you went from 13 variables to 2 variables, and you could do a regression. You could do kind of just some inspection of the biplot, which I'll show you in a minute. So that gives you an idea of the relative importance. Okay. And I guess I just want to show you, since, for sanity's sake, since we did the derivation, and we always do the derivation in terms of the eigenvalue decomposition, but in practice we're using the SVD. So what I want to do here is I want to look at actually find the eigenvalues. I'm going to say here by hand. Obviously, we're not going to do it by hand. I did have to do that once, and that is not a good a fun computation to do on paper. All right, so we have eigenvalues, and I'm going to use the function eigen, okay, which is finding the eigenvalues, and we want to find the eigenvalues of the covariance matrix. That's another command, and we're going to find them for the scaled wine data set, okay. And so we can run this, and this gives back a number of fields. We have the eigenvalues and eigenvectors. Okay, so let's just check, okay? Let's look at the eigenvalues. Okay, so here we have this. Okay, so those are our eigenvalues. And, we can also look at, if I look at names, PCEX3. Okay, let's look at the standard deviations. Okay, oops. Perhaps I did this. Oh, let's look at this. Okay, and actually if we square it, that will give us the variance. Okay, and we will see that, oops, I lost the ability to see the other one, so let me just see. Okay, we will see that they match exactly. Okay, so that's actually a good sign, and that's speaking to the fact that, we can achieve the same thing here. We verified using kind of the doing things by hand that these principal components found using PR comp on using the singular values, we can take the standard deviations from there, square them. We get exactly the eigenvalues, which we know are representative of the variance of the different components. So this is kind of a sanity check. Okay. All right. So I think the last thing I want to show you that relates to principal components is a very important plot called a biplot. And I have not shown you this plot yet, but what you can do is you can use the biplot command, and we can put in our PCA object. And what I want to show you is that, first of all, I want to see PCEX3. Okay, and you know, my screen isn't that big, so let's just do 1 through 5, 1 through 6. Oh dear. I want to show the loadings. Oh dear. Okay. Let's see. Names. Okay, it's the rotation. So the loadings are stored in this object called rotation. And so let's pull the rotation out, and let's just look at a piece of it because we can't see it all on the screen. Okay, so this is the rotation. So these are the different variables as before, and this is the way that they load on the different components. We saw this with the ingredients of the food in the lecture I gave yesterday. Here, PC1, PC2, and PC3. Remember, 0, 0 is kind of like the, we've standardized things, so that's kind of the center point, if you will. And the first principal component is kind of explains the most variability with respect to this total phenols, whatever that is, and malic acid. And then we can kind of look around, and also this ash variable. And then the second one really has to do with alcohol. So we can actually visualize these as a vector. Okay, so for example, if we were to plot each bottle of wine or whatever has a particular profile of scores. Okay, if we plotted those scores, we could also plot these, which are the loadings, and it would give us an idea about the different parts of the plot and how they relate to the different variables. That's actually an attempt of what the biplot does, and I'll talk about this more on Monday when we go into using it for the detection of outliers and other reasons. The biplot is a way to simultaneously view the loadings, which here are denoted as rotation, and also the scores. And the scores are here, X. I believe they are X. Let's see. PCEX3 X. So there's going to be a lot of those. So let's just look at the first bunch. Okay. So in fact, if we do dim, okay, we can see that there's 177 bottles of wine or whatever, 13 principal components, and each one has a score profile. So let's see what this plot looks like. Okay, and it's a really nice plot. And I'll show you this plot again in MATLAB, because here we can kind of zoom in and see really what's going on in the different regions. It's a really nice feature. We can do that using specific packages within MATLAB that are available to you, through UB. Not part of the standard software, but still very useful. So a couple of things that you can see, principal component 1 and principal component 2 are plotted next to each other, and when I do that, it really gets smushed. But these ones, if I were to kind of draw a circle, I would say that these are kind of the ones that are on the outskirts. Okay, they have the most variability with respect to principal component 1 and 2. And we can also see that the loadings plotting the loadings is also very useful. So PC2, for example, I said was really more important for alcohol. Um, and I mentioned some other things about principal component 1. All of those things can be kind of looked at in this light, and we can also see which bottles of wine or which brands of wine kind of align with those loadings. So a very useful plot, and you should do help by plot, and you can see that in addition to looking at the first two, you might look at the first, you know, four different combinations of those. Um, so there's ways to actually input which ones you're interested in looking at. And I believe, okay, they don't give a great, they want us to look at biplot prints comp for examples. Okay, they don't really do a great job with examples, but that's the idea. You have to kind of look at the help file a little bit and figure out how you might plot the different coordinates, but it should be pretty straightforward. Now, the other thing that you might want to do here, which I'm not going to show you. Well, I might show you, or at least give you some fragment of code to look at it by yourself, but you might want to actually draw an ellipse here. And you can do that. You can figure out, you know, the 95% confidence interval of the data in this spread and kind of figure out which the outliers are. And that's a trick we will use, kind of talk about at least on Monday. Okay. Um, I'm going to stop here and I'm going to put the self-organizing map in another little short, a short lecture. Thank you.